thank you all for joining us for this second presentation on mouse models for amyotrophic lateral sclerosis, or ALS, and how to best use them for preclinical drug development. You will see that challenges and new development in ALS research are very similar to the ones described by Katzlert for Friedreich's ataxia. ALS is a severe neurodegenerative disease which leads to loss of locomotion, trouble breathing, and in some cases, cognitive impairment. Many drugs successful in existing mouse models did not work or had only mild benefit in clinical trials. Therefore, there is an increasing interest in developing new mouse models, but also new ways to study them. So we will focus on two powerful biomarkers and show you how we develop them in the lab and how much they can add to the design and the conduct of drug testing. I will focus my presentation on the electromyography, and Max Preza will discuss dipeptide repeats bioassay in C9orf72 ALS models. Let me start by why we are looking for new outcome measures in mouse ALS studies. Over the past 6 years conducting drug efficacy studies at JAX, we came to experience the limitation of traditional assays in detecting the disease manifestation in mice and the disease modification under treatment. And we think that for good drug testing, sensitive assays are needed. And by sensitive, we mean able to detect early symptoms, early onset symptoms, as well as very slight modification after drug administration. And the limitation of traditional measures such as survival or locomotor phenotype is that they interrogate the late stages of the disease. And you'll see example of this. Another requirement for in vivo assays is their reliability. We have all seen differences in results from lab to lab or in the same lab from year to year. And some assays, like locomotor phenotypes, are more prone to inter-experimental variations, and they are also more affected by things that are difficult to control, like animal volitions or stress, or how animals learn about the tasks. And last, the most informative assays are the ones interrogating the molecular or cellular mechanism of the disease and of the drug's mode of actions, because this is what will translate from animal studies to patients. And the issue with monitoring body weight or survival, for instance, is that these are not linked to specific cell type or biochemical pathway. So this consideration led us to develop in the lab the electromyography on live mice to record compound muscle action potential, or CMAP, and repetitive nerve stimulation, or RNS. And this is how it works. So on lightly sedated mice, we record the depolarization of the hind limb muscles, the tibialis anterior or the triceps surae, upon electrical stimulation of the sciatic nerve. This is completely non-invasive and done through the skin, exactly like in the clinic. We adopted to the mice the diagnostic tools used by the clinicians. The depolarization of the muscles, or the CMAP, and you have an example on the middle panel here, is proportional to the number of functional connections between the motor axons and the muscle. The middle panel shows you a healthy CMAP, and the right panel shows you the CMAP recorded in an ALS models, famous SOD1 G93A models developed by Tipu Siddiqui and made available in 1994. And beyond the amplitude of the signal, we can also measure the instability of the signal upon repetitive nerve stimulation. And this instability or decrement will reflect the pathological fatigability of the synapses, and that's also something that is observed in patients. So the fact that this technique is completely non-invasive allows to monitor mice as they progress through disease stages. So on the left panel, you can see how we recorded the same groups of mice SOD1 transgenics in gray and non-carrier controls in black from 40 to 100 days of age. And we found that the CMAP starts to decrease abnormally at about 66 days of age. The instability of the signal, this is the middle panel, starts to increase also abnormally at 66 days of age. And so how does this age of onset compare to the one detected with other assays? On the right panel, I plotted the survival in blue, the body weight in purple, of our ongoing monitoring of SOD1 mice at the Jackson lab. They put the onset at about 120 days for survival, 100 days for body weight. But if you look at other assays like running wheels and rotarod, you spot the onset of the disease at about 75 days. Now you look at electrophysiology, and the onset of the disease appears at about 60 days. That's 1 month earlier than what can be detected, for instance, with a rotarod. And this is just a few days after the onset of the disease could be looked at by histopathology at the neuromuscular junction. And the electrophysiology onset is also visible before the onset of axon loss that you will also see by histopathology. The benefit of the CMAP and the RNS being, of course, that you don't have to euthanize the mice to look at these things. So what are the benefits of the sensitivity of the assay in drug trials? So to look into this, we used the SOD1 transgenics and treated them with a tool compound just after birth, and then we monitored them until 12 weeks of age. We followed healthy non-transgenic mice in green, transgenic mice untreated in red, and transgenic mice treated with the drug in blue. And we used grip strength, the rotarod, and the CMAP, and compared the three outcome measures with time. With the grip strength, we realized that at 9 weeks of age, we were unable to detect any pathology in the untreated mice and, of course, no drug effect. At 12 weeks of age, we could detect the weakness of the untreated mice, but still no drug effect. Moving to the rotarod, we only detected a disease status at 12 weeks and a very mild, significant improvement in treated mice at 12 weeks of age. Now compare this with the electrophysiology that at 9 weeks of age allowed us to already detect the disease in untreated mice and a significant improvement upon treatment. And this trend was even more spectacular at 12 weeks of age, where the disease had progressed, but the drug effect was even more significant. So another advantage of this technique is its reliability. Constant from lab to lab and over the years is the disease onset the way it's spotted. So Seward Woodcoffin here at Harvard Medical School, 2013, spotted the first decrease of the CMAP with the same technique, the same strain at about 9 weeks of age. The curves I've shown you were recorded at JAX early on in 2014. In 2018, we were still recording the same age of onset at 9 weeks of age. And 2 years ago, a group showing the role of ASOs in correcting the disease in the SOD1 mice also detected the onset of the disease at 9 weeks of age. So finally, the benefit of the EMG for animal studies that we became aware of and I wanted to share with you is that as more and more disease-associated variants are discovered in patients, more and more mouse models become available. How do we know which one will be clinically relevant and beneficial for drug studies? So in the instance I'm showing you here, we reproduce in the mouse a mutation associated with ALS in the NEC1 gene. So the finding was from John Landers, and we generated CRISPR mice at JAX. The homozygote mice looked very promising at first. They were small, shaky, they had tremor, they had trouble walking. And so we thought we had developed a wonderful new ALS model. However, the homozygote mice had a relatively healthy CMAP. So did the heterozygote mice. And if anything, their CMAPs were just slightly higher than in wild-type control mice. So they did not have the peripheral neuropathy that you expect from an ALS model. And that was confirmed when we looked at their nerves by histology. They had absolutely no nerve deficit. So the EMG can also serve here to choose models with clinically relevant features. So not all mouse models will develop EMG phenotypes, though. So that's especially true for current mouse models for ALS caused by mutation in C9 or 72. That doesn't mean that these mice don't have clinically relevant phenotype, but this will lead us to another biomarker, biochemical, this one, and this is what Max is going to tell you about.
Hi. Hello. Thank you for this chance to talk. I'm going to change now. Okay. So I wanted to talk a bit about different mouse models that we are working here at JAX, and it's related to the C9orf72 gene and the relationship with ALS pathology. So C9 is perhaps the most commonly mutated gene in familial ALS and in ALS patients in general. The most common mutation consists in an expansion of an hexanucleotide repeat from 20 to 30, which can be normally found in healthy people, to more than 100 in ALS patients. So the consequences of this expansion, they translate in pathology to multiple mechanisms. One of them is produced by the generation of transcripts that can be accumulated in the nucleus and form RNA foci, which can also accumulate and sequester nucleoproteins. But also, some of these aberrant transcripts can reach the cytoplasm and get translated in a particular non-conventional method called a repeat-associated non-AUG translation and initiation RAM peptides, what we call it. So this generates an array of 5 different dipeptides, polydipeptides, consequences of the translation in all the different reading frames. So these dipeptide proteins are highly toxic, and they can get accumulated in the cytoplasm, interact with another RNA and other proteins, and produce aggregates and insoluble aggregates that are toxic to neurons and cause neural degeneration. So today I wanted to introduce a new mouse model that we are using in the lab. It was developed first by Dr. Leonard Petruccelli's lab, and it consists basically in induce the overexpression of the C9 or RF72 repeats with expanded repeat. In this case, we are using an AV9 vector that carries 149 repeats of the hexanucleotide from C9, and also a control AV9 vector that carries only 2 repeats. So the model consists in inject a third at postnatal day 0, black 6, 657 black 6 mice via ICV, and then monitor the mice for survival, growth, and behavior at different time points after injections. So here in the lab, we were able to reproduce most of the findings that Dr. Petruccelli found in his lab, like the most important neurodegeneration, DPR pathology, TP43 pathology, and also gliosis and astrocytosis. So one of the findings that I want to share today is how poly-GP DPRs can be detected in 149R injected mice. This is also in agreement with the findings from the original paper from Dr. Petruccelli. But we saw here is an increase in detection of DPRs, poly-GP, and it's also probably age-related. You can also observe how is highly specific. We don't see almost nothing in mice, control mice injected with with AV9-2R repeats. Now, if we want to use the abundance of poly-GP as a good biomarker or reliable biomarker, what we need is more high throughput assay and something more quantitative. So we work at a method based on previous publications. This method is based on the use of a mesoscale discovery technology, and basically consists in use an antibody against specific for poly-GP peptides. Basically, what we do is generate protein lysates from frozen brains, and then use the detergent soluble fraction to quantify through this immunoassay the amount or the abundance of poly-GP. In this case, it's the soluble fraction that we have optimized. So what we do is incubate this lysate in the presence of a capture antibody and then a detector antibody, and then we can run this like a regular ELISA through the MSD immunoassay. By now, we have this method fairly optimized for the detection of poly-GP in the soluble fraction, as you see here. And the other aspect to this to highlight is the high speed and easy performance from this assay. So what I'm showing here is what we have from this validation tests for analysis for the AAV9 model. So we run brain samples from mice dissected at 6 months, 9 months, and 12 months. And we were able to run the assay, and as you see, the assay is really highly specific. The poly-GP DPRs are only detected in mice injected with the AAV9 149 repeat, not in the AAV2 repeat, the control AAV. Something unexpected was that actually there is an age dependency, but what we saw in the analysis for soluble fractions is actually a decrease with age. It's kind of an unexpected result, but we are trying to work different possibilities to answer this result. One of them could be that we actually, since we have neurodegeneration, it could be possible that some of the neurons that were transduced with the AAV are actually dying. That will explain a decrease in the production of soluble poly-GP. And the other possibility will be that actually the poly-GP DPRs are accumulating in insoluble fraction that increases with the age. So that's another aspect that we are actually working hard to solve. And finally, what I wanted to share here to the left is an example of a clear application of this method, where a control mouse, which is here as reference, is a mouse injected with AAV149R, and then used as reference for the analysis of the effect of two different drugs targeting C9 or S72 repeats. And you can see how this will be a clearly an easy application for this method. So our future directions will be first try to include the analysis of many of the C9 or F72 transgenic mice models that we have here at JAX. We have a very large panel of mice, and it's a really interesting model that we should be able to assess through these assays. And also we want to optimize the use and the analysis of the detergent insoluble fraction for future analysis. And so as conclusions from this Laurent and my talk, we wanted to say that in vivo assays like CMAP measurements are very sensitive in vivo assays to detect neurological phenotypes and at onset and modification after several treatments. In terms of biomarkers, DPRs are very sensitive biomarkers, and it could be a new assay that being a game changer in quantitative and high throughput drug testing for C9 ALS. So if we take together all these assays, interrogate the specific cells affected by ALS and targeted by therapeutics. So these new biomarkers-based assays we think are superior traditional assays and rely only in general modification of behavior, clinical observation of lifetime. I want to thank you, and I'll be happy to answer any questions.